Hello and welcome to another video tutorial for BIOS E1A. In this tutorial we'll work through a practice problem concerned with amino acids, properties of their side chains, or R groups, and the effects of amino acid substitutions on protein structure. The particular context for this practice problem is sickle cell disease and hemoglobin. Before we get to the answers, I'll begin with a little background info. You may know a bit about hemoglobin already. Hemoglobin is a protein, as you know from lecture and the textbook. That means it's a macromolecule made up of amino acids. Hemoglobin protein is found in red blood cells where it transports oxygen. In this problem, we'll be talking about effects of amino acid substitutions on protein structure. Hemoglobin is a tetramer, meaning it's a protein composed of 4 subunits, each of those subunits being a polypeptide. 2 of the polypeptide subunits of hemoglobin are alpha chains, while the other 2 subunits of the major form of hemoglobin in adults are beta chains. As you learned from lecture, this also means that hemoglobin has quaternary structure. It's a protein comprising multiple separate polypeptide chains. In normal hemoglobin protein, the 6th amino acid in one beta chain is the amino acid glutamate, or glutamic acid, depicted here with a pale red circle. This is the hemoglobin in normal red blood cells. In sickle cell disease, however, hemoglobin is altered. Instead of a glutamic acid, sickle cell hemoglobin has substituted another amino acid, valine. Under certain low oxygen conditions, the sickle cell variant of hemoglobin can aggregate into fibers, causing the red blood cells to take on a sickle morphology and leading to sickle cell disease. Now let's come back from background info to the original problem. Starting with part A and using what you've learned from lecture about different amino acids and their R groups, how can we explain why changing glutamate to valine might affect hemoglobin structure and function in this way? Remember that what distinguishes the amino acids from one another are the particular characteristics of their R groups, or their variable side chains. The 20 amino acids can be grouped based on their side chains. Some have positively or negatively charged hydrophilic R groups. Others are hydrophilic and polar but uncharged, and others have nonpolar and hydrophobic R groups. Normal hemoglobin has glutamate at position 6 of its beta chain. We see, if we look at glutamate or glutamic acid in the upper right-hand corner of the amino acid chart, that glutamic acid has a negatively charged R group. Sickle cell hemoglobin instead has a valine, which we see, finding it at the bottom right corner of the chart, is an amino acid with a nonpolar hydrophobic side chain. So now we can answer part A. We know that the glutamate to valine substitution exchanges amino acids with very different R groups. One, glutamate, is charged and hydrophilic. The other, valine, is nonpolar and hydrophobic. This substitution thus significantly impacts the chemical interactions possible at residue 6 and alters hemoglobin function. We can reason in the opposite direction to answer part B. Substitution of an amino acid with a very different R group altered hemoglobin function. So, if our goal is to make a substitution without impairing function, we'd want to pick an amino acid with very similar R group. Let's refer back to the amino acid chart in our original beta chain position 6 glutamate. The most similar substitute would be aspartate, or aspartic acid. Its R group is also negatively charged, and in fact it only differs from glutamate by its side chain being 1 CH2 shorter. Other good choices of amino acids with similar R groups would be asparagine and glutamine. Their R groups don't end with a carboxyl group, and they are uncharged instead of negatively charged. However, their side chains are similar in size and are hydrophilic and polar like glutamate. If glutamate was, for example, mainly interacting with water molecules, polar asparagine and glutamine would be good substitutes. To summarize, for part B, like-to-like substitution of amino acids with the most similar R groups gives the best chance to avoid impairing protein function. In this case, these substitutes being aspartic acid first, and then asparagine and glutamine. We can extend our reasoning from part B then to handle part C of this practice problem. Now we can understand how you could make substitutions at one or multiple residues without significantly changing protein structure and function. The like-for-like substitution of amino acids with very similar R groups at one or multiple residues would create two proteins with different sequence at those residues but similar structure. For example, we can imagine hypothetical protein A with a leucine at position 3, highlighted in green here. We can then imagine a protein B with another nonpolar amino acid like alanine, valine, or isoleucine, whose structure and function would be very similar. And we can imagine like-for-like substitutions at many other residue positions, too, such as swapping positively charged lysine, here in blue, for another positively charged amino acid, arginine, leading to two proteins with many differences but very similar structure. That concludes this tutorial for BIOS E1A. Remember, if you have additional questions, you can post them to the course discussion forums, or of course bring them up at weekly discussion section office hours or via email. Thank you very much again for listening.